Hello,
Hello.
Professor. How are you?
Hello. Good. And you?
Yeah, same here, same here. Thank you for
Uh,
accepting our talk, for invitation. Appreciate.
yeah, great.
Um,
Very thanks for the invitations. Okay, here, how about over there, the weather?
the weather is very nice. Is that Golden Gate in the background?
Ah, yes, right. So actually, this is 7 a.m. in the morning.
Yeah, thank
So,
you so much. Thank you so much for
okay, no
accommodating us.
problem.
Do you want to share your front page to see?
Okay, yes. So you allowed me to share the slide.
John, there's John Gozal. That's his.
Okay,
Yes, you should be allowed to share. Let me
okay.
just check. Uh, yes. So we see you sharing. Okay. Yeah.
Okay. Hi. Okay. Date is long. So I think it's your. I little bit mixed up because your day is March 11. Our day is March 12.
Oh.
So okay, makes thanks. Sorry for that. Okay.
No problem, no problem. John, I see left bottom. There's a person.
Yes, yeah. That looks like an
It
artifact.
disappeared. Oh, it's still there now. I see myself there.
I think if you turn off your camera, Mehmet, it might go away. Mm, hmm. This is something new. I think Professor Choi, can you try starting over the screen share and only share your PowerPoint rather than your full screen? I think that might solve the issue.
Um.
It looks like this might be an iPad Pro or something that you're using.
Yeah. Right. So. Uh, that is,
Right? If there's a way to only select the PowerPoint app.
okay. Hmm. Hmm. Oh, I don't know how to put the.
Yeah, it seems like it's just an artifact of the iPad Pro Zoom. Maybe if you can just,
Oh,
oh,
okay.
I think that'll
I
do it.
shifting. I shifting. Yeah. Okay.
All right, looks good.
Excellent. Excellent. So, Professor Cho, the way we do it, we wait 3 minutes after the hour, and then I introduce you. You have the podium 45 to 50 minutes, and then I read questions from the chat box. I hope that's okay.
Okay. So you're gonna give it 50 minutes.
Yes, whatever you have, 45, 50, 55, you know,
Oh yeah. I'm expecting 40 minutes.
that's fine too. That's fine too. Your work is excellent. So we will take that one too, no problem.
Okay. Thank you.
By the way, Abdullah is doing excellent work here. Appreciate your training him and mentoring him.
Oh, great. He's doing very well. Yeah.
Yes, yes.
Yeah. He was actually. You already knew that he was great jobs he did, and he published a lot of papers. And then, you know, he's very responsibility and if assigned any kind of a task, he successfully did. And very, confident guys. Hi, and actually, I'm attending Bio USA in San Diego.
Okay.
June, I think, to some. So I'm gonna visit and Terasaki Institute because on the way back to Korea, I dropped LA.
Okay.
So yeah, just allow me to.
Sure. Anytime. Let us know. Let
visit.
Abdul know. Let me know. We will organize you to meet couple people at the institute. Shilling also wanted to meet with you. About your interesting research. Definitely. Just let me know the dates.
Yes, great. Dr. Abdul, please, yes, informed my schedule.
Excellent. So
Okay.
it's 3:05. Let me get started on the talk. Good
Yes.
afternoon, everyone. Today we have Professor Cho from Jeju National University. He was previously led the biomechatronics laboratory. He has served as a visiting professor at the University of Tokyo, University of Waterloo, and the University of Twente. He received his PhD from the University of Ottawa. His research focuses on organs on a chip and microphysiological systems, as well as AI-driven antibody drug conjugate development. He has supervised more than 80 master's and PhD students, published over 300 peer-reviewed journal articles, and holds more than 80 patents. He is the founder of Biosphero, a company specializing in organs on a chip platforms, and currently serves as the CTO of Biologics for You, an AI-based ADC technology company. The floor is yours, Professor.
Okay, thank you. Thank you very much for the kind of introductions. I would be likely sincerely thanks the Terasaki Institute for inviting me to speak in the webinar today. It's a great pleasure and honor to be here and share our work with you. Hi, my name is Hyun Choi. I'm a founder and CTO of Biospero and Biologics for You at the Professor Imisha of Jeju National University. Today, I'm pleased to share the research result of the both companies. In the first part of the talk, I will introduce OrganXpert and our next generation microphysiological system platforms for human relevance testing. In second part, I will show how we are connecting this platform with AI-driven ADC development for specific liver cancers. Through this work, we are aiming to bridge in silicon discovery with human relevant validation supports better drug development and create strong opportunities for the collaboration. Let me start OrganXpert TMs. Okay, microphysiological systems, I'll let you know, are becoming increasingly important because they can replace the human microenvironment and organ level structures functions more closely than conventional in vitro model like 2D or organoids. As you see in one of the slide, the main biological idea is to recreate the key issues interface, especially the epithelium and endotheliums within the microfluidic chip platforms. This allows us to build a more human relevant model for organs, such as the good liver, lung, and kidney, and oncology, and to evaluate both toxicity and efficacy in a more realistic settings. MPS also has broad applications. It's relevant to the not only for the drug development, but also for personalized medicine or precision medicine and cosmetic many other areas. So here, MPS is not a narrow research tool, but a broad enabling platforms for the next generation tasks. Why MPS matters? Actually, animal models remain widely used currently, but their predictive values for the human outcomes is very limited. In these comparisons, animal in vivo models are at the very lowest and almost zero. 3D have set spheroid improved prediction to around 47%, and liver on a chip research is about 87 prediction accuracy for the hepatotoxicity drugs. Better prediction, so based on this one, a better prediction can help prevent the failures before the clinical stage, improve regulatory relevance, and reduce human harms from unexpected adverse drug reactions. This is why they are global pharmaceutical company are rapidly adopting MPS to reduce risk, cost, and the time market. Now, the scientific need for the MPS is very strong, actually more than 20, 30 years. But the regulatory environment is also changing nowadays in very important ways. The global market is moving toward human relevant testing platforms in the United States. The FDA Moderate Act 2.10 signed that the animal testing is no longer mandatory for the IND-related submissions. Also, last year, April, also FDA announced that the roadmap of the animal testing free, specifically antibody therapeutics, antibody-related some researches. Also in the EU, there is a growing adoption of NAMs, and Japan and Korea, there are ongoing effort to integrate MPS in vitro model into drug safety evaluation framework. So this is not only a scientific trend, if also regulatory and market transitions. We believe that Biospero and your research result are well positioned to shift with the platforms that are generating more human relevant data. This is a very helpful slide and shows the practical impact of the organ on a chip in drug development. Today, the drug development still take about 13 years, costs around $2 billion, and have a failure rate close to 90%. But the 90% is about the oncology aspect. Some experts and researchers announced that fail rate is 70.5, only 2.5 success rate. It's very less success rate. So this chart is on the right side shows the major causes of failure, including lack of clinical efficacy, poor drug-like properties, and unresolved toxicity and lack of the commercial lead. The key message is that here, organ-on-a-chip models can enable earlier detection of efficacy toxicity, which helps reduce time, cost, failures. The slide also noted that around 80% of these issues could potentially reduced by organ-on-a-chip models. So when we say organ-on-a-chip can can fail in the opportunity. We mean that it can help authors to make a better decision earlier before too much time and money are spent. So let me introduce the OrganXpert systems. That is our company. Our company major product is the value of proportion of the organ on a chip. This one is a very clear OrganXpert to provide new paradigms in drug development by combining high throughput operation, bird's-eye organ models, and real-time physiological relevance. In other words, this is not only chip, it is a practical testing platform. It can run the 12 chips in parallel with right now independent control. It has built incubator for stable long-term cultures. It supports multiple organs and disease model for efficacy and toxicity studies. Its advanced perfusion systems mimics real blood fluid and a tissue exposure. It supports epithelium and endothelium cockroaches for barrier and immune studies. The customers can get benefit. OrganXpert also clearly shows a faster and more predictive preclinical testing reduce costs. So actually we got some award, design award, global award. And here on detailed explanation of that consists of the three models. So here, interesting one is OrganXpert is already I expressed that it's a recirculation-based fluidic systems and built-in incubator, and also it will be UV sterilization. Organ O is a co-organ barrier on the chips. So we can build any kind of model with OrganXpert O. However, many researchers still find organ on a chip difficult to utilize that. To solve this problem, we developed a tool help the researchers who are more familiar with 2D cultures and to build 3D tissue models more easily and comfortably. This is Organ T is a tool for the basic cell studies, especially co-cultures and stainings. So within this complete connected platform families, we can support the basic studies or advanced organ models and the highest throughput palm workflows. I will introduce to show the videos, the OrganXpert.
BioSparrow's organ-on-a-chip system is an all-in-one platform that replicates the microenvironment of human organs and tissues, positioning us as leaders in the field. The BioSparrow's organ-on-a-chip system consists of 3 key parts. Our OrganXpert O chip uses human cells to evaluate drug efficacy and toxicity, emerging as a powerful alternative to animal testing. Its core features, the multi-layer structure and circulation system, mimic major human organs like the liver, kidneys, and lungs, replicating blood flow and creating conditions similar to the human body. This greatly enhances both accuracy and reliability. Here's how the experimental process works with our organ-on-a-chip system. First, human-derived cells are cultured in the OrganXpert T. The cells are monitored in real time and then connected to the OrganXpert O. Once connected, the cells receive the necessary nutrients and drugs through the circulation system of OrganXpert O. The experiment is conducted in the OrganXpert M microphysiological system, which connects up to 12 organ chips at once, minimizing errors and delivering accurate results. Our easy-to-use cartridge system prevents contamination and ensures seamless operation, another innovation unique to BioSparrow. By integrating a microphysiological environment module, we create optimal conditions for experiments and deliver fast, accurate data. BioSparrow's organ-on-a-chip system supports the cultivation and testing of human organ cells and can be applied across a wide range of fields, from drug development to regenerative medicine.
Okay, this is introduce the Okay, this is a coming soon automated high throughput organ-on-a-chip with the AI powered. Actually, previous one was for the research groups. However, it's many groups are repeatability issues, and also the planning and the perceptions, automate monitoring, online monitoring, and also if we got some data and we need some analyzed for the different with the multi-omics data. So we are implementing those perception and activation and analysis with the AI, and also we encouraging automate the high throughput organ-on-a-chip platforms, real-time monitoring sensors, robot driving media, and handling the changing, sampling, some any kind of the human beings, the operations. Okay, so this is not just the next hardware presence. Okay, it's our vision for the more intelligent, scalable organ-on-a-chips infrastructures. So for collaborations, though, we are open opportunities only in biological, but also sensing and automations. So AI analysis, high throughput screening that we do. Actually, in our systems previously, we include some biosensors like lactator, glucoses, and TR and ROS with electrochemical sensors. And also that another one is the TR sensors. By using TR sensors, also we are working on the optical sensors. So those systems, sensor systems, will be implemented there. Okay, let me try to explain briefly some result. This is a liver on a chip. So we build a model for the livers, iPS-derived that 4 different of the cells, hepatocyte, fibrous Cooper cells, endothelium cells, and then the systems multi cells under a fluid which allows more realistic liver microenvironment. Actually say that in the system say that circulation, like human beings. Also important one is can be shared stresses. So our research group can control shared stresses. So shared stress is important to the phenotypes, express phenotypes. So we are validation using the FDA approved drugs, moderate and severe and mild. And the blinding test that we get some AST evidence of the changing in albumin and production. Also because in organ-on-a-chips can there's 3 kind of omics data. They can transcriptomics, proteomics, and metabolic. So those are kind of the data that can be measured by assay systems, right? So is it possible to genomics and therapeutics combine that is more precision drugs developed for the patient. Actually, we have initiating several projects with Japan. They are one company is working for some genomics based therapeutics for the cancers. They want to do the other 3 omics data, and those one can be using, high complexity and very relevant to human beings systems. So they are updating some precision drugs for the therapeutics purposes. The other one in the Einstein on a chips. This one is interesting on a chips. It's application drug, it's permeability and absorption studies, because our system is a bilayer system, right? So it's a barriers there. That's why it's a liver and intestine and lung, or also kidney and oncology, and right, those are kind of organs very strongly we can contribute to those kind of the organs. So we show that a barrier formation by GO1 staining by 7 days, tight junctions maturation rate indication successive establishment in intestine epithelium and barrier functions. So actually, our systems platform is not screen proposed. So very wide spectrum from the drug development stage is very early stage screening, but our system is very high complexity. So our spectrum is near the IND. Okay, so it's a very with the connecting IBIBE, so those data we are supporting IBIBE as tools compared to the 2D, IBIBE is not successful working that way. So these data is very close result clinical levels. So that's why our spectrum is very close IND. So previously I mentioning that the automatic systems, and then it can be shifted to the IND level and shift to the screen levels. So we covered a wider range of the area of the spectrums. Also, this instance, this one is hypoxia stress mechanism that show CACO2 and the Hubeck-based Einstein Chi model. So here the HIF alpha images confirm that the systems respond biologically under hypoxia conditions. The viability imaging graph shows the decreased viability under hypoxia conditions, the permeability and result confirm the barrier breakdown with increased FITC dextran leakage. So we can, the barrier tight junctions can be modeled and can be evaluated by our, model systems. So this can be said that the model can reproduce not only a normal barrier formation, but also pathological stress conditions such as hypoxia-related barrier dysfunctions. Actually, so this system can be used, this microphysiological system even covered the pathological, model. Kidney model. Kidney model is also very, we can contribute very strong to use the barrier. So here say that how TJ-beta is induced, fibrotic kidney state, including, reduced capillary density, stiff extracellular matrix, reduced perfusion, increased hypoxia. So the kidney fibrosis model is designed to capture these changes. So there is show the decreased KIF8, increase alpha SMA after TGF-beta treatment. That's the model molecular and validation graph show that sick vessel decrease and the syn vessel increase and the fibrosis which inhibit the treatment, reverse this trend. So main result is that kidney on a chip platforms can reproduce both structure and molecular features of renal fibrosis. And we applied that. We are AI-based repurposing drugs for the fibrosis. So we find the ruby stones as an example derived from AI repurposing tool. So presented and tube interstitial fibrosis, the proposed model of axon ruby proteins and the kidney fibrosis chip model. And also we did testing with the animal tests. So if you compare that, PCR is very closely related that we published the papers. And we say that we can say that the chip can detect meaningful anti-fibrosis drug responses, both imaging and molecular level. The markers are useful not only for the decrease modeling, but also absent using the drug screening. And skin on a chip also we are currently collaborating with a company in Japan. They are wanting to full, skin on a chip with endothelium cells. So the important one is the full scale, 1 millimeters per thickness, and they're gonna, application to the different drugs applications. So this one can show that relevant way than the static cultures. This is a values of skin biology and dermatology barrier studies and cosmetic testings. So I say that the endothelium layers, it is also possible to build a full skin model with a basket. So we have some R&D centers, manufacturing facility, and marketing. We have trying to harvard by sparrows, and now we build a Korean new, clinical CRO and university hospital, and then some, company, pharmaceutical company. So we're gonna be extending to the global stage. So we have the many different, companies and in the Japan and TNO in Netherlands and United States to a children's hospital LA. And there are to Europe, the company we are, connected and do some researches developing some package based. Okay, now I am shifting to the second part of my talk, which is focusing on ADC development. So we have buildings overview of a full ADC development workflows. We have ADC GenX and Replica GenX. We do target antigen-based antibody GenX and a payload GenX and a linker GenX. So the novel ADC we developed and we validated Replica GenX with the organ-on-a chips. In a very briefly overview of AD GenX, you can see that all of them AI-driven systems, platforms, it's antibody GenX and payload GenX as a link. So this is connected 3 of the modules and developed the de novo ADC. So we are not, working on the synthesize. So we are connecting with some, reliable CDMO globally like AppGena. There are many, collaborators. So we are synthesized, conjugated from those company, and then we'll do Replica GenX. Okay, ADC GenX, payload GenX is to 2 billion molecules. From that, we are screening that and 4 different MOA we have. For example, in DNA damaging microtubules or topo-1 or degraded. So we are based on the MOA based. We are suggested at least 5, and then in the evaluated in silico levels. All the developability and cytotoxicity, all the parameters can be evaluated in the in silico and the synthesized. We do our testing. So we developed the microtubule. We developed right now is also microtubule for the livers, and we are developing topo-1 and degraded. And the linker GenX, the ADC linker GenX linker is very important. We are by using the converters generated 101,000 linker candidate using a small format. We are learnings based on those one. And we developed the new de novo linkers. It's not easy, but we anyway, we developed that. We synthesized with conjugated with the payload. We have that one. So and now we have a platforms, linker and a payload. The payload will be different, right? Depends on the applications. Can be DNA damage or topo-1 or degrade for the DAC. And now this platform is totally independent of the antibodies. Antibody GenX, this is, we are focusing on bioparatopic or biospecific and site-specific antibody. So currently we do our liver targeting GPC3 and CD90, right now EMPPPA3. There are targeting, and we analyze that antigen epitope residue, and we are finding proper residue waiving free over the patent. So we design that and we synthesize, and also we are testing that. Also nano body, also we have the platforms for design nano bodies as well. So because cancer, TME is solid cancer, is the penetration issues are, so we are trying to the, different methods, like peptide substance, but nanobody is one of the solutions because very less, molecular weight. Okay, we fully developed the bispecific, biparatopic site-specific ADC for the liver cancers, GPC. So we find the 2 different paratopics. We have the linkers and we have the payloads. So we are developing, GPC3 biparatopic antibody. So we are doing some testing right now, and the development is going to be June or July. So we're going to introduce that. This company is the platform company for asset for the pre-IND package to the licensing out. So how about we applied some organ-on-a-chip for the development stage? We need some confirmation and confidence our system is working for the ADC. We are employed the Kadcyla is already approved drug, the ADC drugs. So we do some testing here, internalization, penetration, some efficacies, and compare to because the Kadcyla already proven drugs, and there are many informations, biological information. We are, hepatotoxicities and internalizations, efficacy. We do tests, it's very similar, result. Okay. And then also we do some, permeability testing, other models. TME model is very important, right? So validation of Kadacilla efficacy, the breast cancer on a chips we do. Also, there is apoptosis, breast cancer on a chip internalization, and the killing that. Also now that was actually the purpose of that is our replica Gen X is applicable to the ADC validations. So we did using Kadacilla based. So now we are developing liver cancers. So we build the liver cancers on the here we are checking the expression of that and dynamic conditions. We do some GPC3 expression, the Ki69 alpha SMA. So ATF, that is expressions we do. So GPC3 and we are characterizations, you can see that liver cancer ADC binding affinity internalizations, and those kind of the testing we do with the ADC. Okay. Thank you so much. I explained to different kind of the researches, but I say that this is not totally different. Because first OrganXpert provide human relevant MPS or organ-on-a-chip platform that in a sport toxicity efficacy and the disease modeling across the multiple organs. And the second one, ADC Gen X and replica Gen X connect the AI-driven ADC design with the non-oncology as well is with the human-centric chip-based validation. So we believe that this integrated approaches can improve preclinical decision-making, create among strong opportunities for the collaborative development. Okay, thank you so much for your attention. I would be very happy to continue the discussion and explore possible collaboration opportunities. Thank you very much.
Thank you, Professor, for the nice seminar and the extensive data you showed. Many questions came in. One is, how reliably does this microphysiological chip recreate the blood-brain barrier?
Okay, sorry for another question. Once again.
How reliably does your MPS recreate the blood-brain barrier?
So you mean brain or barrier or
blood-brain? The vasculature in the brain, like what is the tear value?
BBB.
Yeah,
right?
BBB.
BBB.
Yes.
Yeah, why? Okay, yes, and I didn't show the BBB. Actually, BBB is our main focus. Okay, so we built the BBB model, and we check the two kind of tight junction. One is the TER sensors. Another one is the DAX side, staining purpose. Currently, we are build that. Delivered to the Seattle company. Seattle company will do some service with the BBB model. So we tested several BBB using some solutions, and we find out some tight junctions, and before and after tight junctions with that. So BBB is actually very nice, system, because this is bi-layer chips, right? So bottom endothelium and bottom the epithelium cell side, and very close to the human, BBB model.
Thank you. Second question is, as an expert in the organs on a chip field, can you please provide your perspective about the best part of the studies using organs on a chip platform compared to using conventional organoids?
Yes, good questions. Very good questions. As you know, already in the United States, FDA is very prepared the organochips instead of the organoids. The main thing the organoid is repeatability. The very different groups shows a different result as well. So it's a repeatability is very long and not match the stages of organoids. So organ-on-a-chip case in already many years, and also is a numerical values based data. So organoid is very qualitative result is that, and organ-on-a-chip is very quantitative information provided that. Also organ-on-a-chip case and very similar or close to the structures of a tissue models, and also there is some easy to mimics of the human microenvironment like circulations, and also we can check some biomarkers. But organoid is very limited, for different multi-omics data as can that. But our systems also including some organoids in our systems. We do some testing proposed for but it's good. Organoid is growth and is good. However, compared to organ-on-a-chip, evaluation of drugs definitely very different. I cannot say that the other different technology. I don't want to tackle that in the liver testing, right? I say 3 different drugs we tested. Actually, we did 4 different groups we do. We are organ-on-a-chip result, and the other group 2D and animal testing, and the organoid group. The 3 groups doesn't have the good results, and then that's why we did not publish the papers, because those groups are, hesitate to do some, collaboration results. So yeah, definitely I can say that. Yeah, organ-on-a-chip is a very mature stage, showing some any kind of information for FDA requirement, but organoids need more studies actually.
Thank you. For the ADC screening, what is the most important part that we need to consider? Of course, the target antigen and payload will be parts of important factors, but is there anything more than we need to consider for the ADC generation?
Okay, actually our linker, the linker you have, your own linkers, right? There are many linkers. Antibody is also important. However, the most really important one is the payload. Payload should be a novel payload, new payload, or I say a very different, MOA there. The currently microtubule instead of the top one is now right trend. That's because we analyze the several, thousands trying data. So top one is also, new trend. So a linker and a linkage you have already, and a payload is actually key issues. And the linker is connected because the payload and linker is independent of the antibody. So it's connect the site specifics. Right? That's why I say payload is very important. So you can say that right now monoclonal and single payload is not acceptable right now currently in the market. You have to site specific or bispecific and a dual payload. So if you have the payload that, and you can do some dual payload, and there are many different, or degraders. So you can say that Protox, DAC, and RPP, so peptide based, conjugations, so extendable bedding.
Thank you. A follow-up question. You mentioned that conventional organoids have batch-to-batch variations, which means they have different maturation levels. Does it mean organs-on-a-chip platforms provide less batch-to-batch variations?
Definitely. But that's what I'm saying. Currently, a conventional organ on a chip is very totally dependent on the researchers. So that it means human errors involved with that. So even the same, your groups, the one guy and the other guys has showing the different results because their skills depends on their skills. So that's why we are providing, try to removing the human errors that are, kits, and to try repeatability, no variations. That is our strong point. And next version is also the removal of the whole human beings, also human beings, by using the robot systems and AI using that. Removing human beings operation analysis. Also, analysis is a very different approaches right now. You have a lot of assay data is coming from the assay is a transcriptomics or proteomics or metabolomics. Those one can be very integrated but heterogeneous data, right? So it depends on your expertise that you can analyze that, but it's not good. Approaches that clinical stage is very complicated that you are based on those heterogeneous data should be analyzed by using multi-omics integration AI tools. So those tools and system hardware also is a monitoring system, right? 7 days you're working on that, you have to decide to go or not. So currently is a black box based. So after you are 7 days later, you are very exciting and nervous to result, but the result is not acceptable. You spend 7 days, right, to money. But for preventing that, so online monitoring should be there. So I say white box, to say. So we can decide that, to go or not. Also, you can see that in the dinner time or your houses through the smartphones, when your system is going on. So if drugs treatment, the result is going very smoothly goes. So you want to be happy. But you applied some drugs, no efficacy trends or signals, and next stage you can go and stop that one. So online monitoring is very helpful to the organ on a chip models and organ on a chip evaluations.
Thank you. Could you please describe some regenerative medicine applications of your OrganXpert MPS platform? It would be helpful if you could share a few examples where this platform has been evaluated if this platform is evaluated in regenerative medicine, like tissue engineering.
Okay, so currently, because we cannot do all kind of the model, but however, currently we are initiating some collaborators right now. So Seoul Bundang University, Hospital University, they are regenerative the medicines right now. Their workings. So it's a very exciting areas right now. Also, we are in Abu Dhabi. They are suggesting some proposal with together for the regenerative medicines. Okay, so actually organ on a chip, this kind of system, not a simple one. These systems can be very complex, high complexity, very mimics, right? So this is a very proper approach to regenerative the medicines. Okay, also we are working on some company is we are organ on a chip, and the back of the endpoint, they have on systems, they are metabolicity assay kits like that, right? So for together combined the package, we're going to do some business that one. So for the specifically. precision or regenerative personalized medicines, we apply that one. So basically, mostly, I say Japan mostly say that cancers. Right now, we are focusing on lung cancers right now. So they are shifting to the different one that is renal cell cancers.
Thank you. Regarding your OrganXpert platform, have you integrated any sensors beside tiers such as antibody aptamer-based biosensors for albumin detection or other things?
Good questions. Actually, in current systems, not including sensors. So previous version, we do TR, ROS, oxygen sense, and lactator, glucoses, or antibody, APT. There are different antibody sensors we do. Okay, so however, when we develop the new models, so there is some technical issues, the position and the structure, the design. So we are holding that one. But however, next version I showed that is including those sensors. I can suggest to you that, okay, there I see already mentioning that there are different biosensor technology and approaches. First one is the TR. The second one is chemical electrochemical sensor. Electrochemical sensors provide the signals very nicely, pattern-based. So you can do enzyme-based, you can do that, or molecular-based do. And also you can do antibody-based that. So that can be easily integrated that, I don't know, or you are, current organ on a chip. I heard that you developed many good organ on the chips that is possibly integrated that. So those three kind of the approaches that also more you can say the optical as well. So optical is including 4 types of different one can be covered all over the organ on the chips lead out.
Thank you. One other question, is your company providing a contract research service, or are you selling this machine, and how much is it?
Okay, yes, actually, currently, basically approaches are service as well, but basically focusing on right now, selling that. So for example, the collaboration approach is that if you have, very, for example, example is better. Children's Hospital LA, there is a kidney. They say many kind of kidney, cell types cells. So they're gonna provide and building some disease and also their research purpose or testing. So they do. In our system, they build, the models, and then those chips will sell them. That is our, collaboration with them. That is our next version of that. Because if you have some, Terasaki for example, pancreas, models you develop. So with our, chips. And then those chips, including tissue models, and we can sell them, or you can sell them together. So the client, final client only need, systems, platform, and the chip is provided by Terasaki Institute, right? It's including all the model. So their job is do not need to build this, pancreas model. Instead of that, they only need drug testing. So they can save the time and the money because they do not need any biological research to building some model, right? So also the model is Terasaki Institute build the model, the very highly reliable, expertise-based. Client believe that, very confident that. So actually, that's why I'm saying is one company in the United States developing some antibody therapeutics. He said he want like this one right now, but the problem is the cold chains. We developed 3 or 5 days, sustainable model. We do research right now. If you have a building that within 5 days, and they do without any loss of the functions of the physiological or any kind of the phenotypes without losses, you can provide that. Okay, so I say that the 3 kind of business. One is we're selling platforms, the platform. Second one is with the sales, it's model selling, and then like services. That is the price is that more in the incubator, in the putting incubators. It's standard model is by $80,000. And the pro model is incubating a function is including that is $100,000 for that. And then the chip is consumable. That is our current prices. So, but we can, Terasaki and collaborate with the authors, and we are providing very less prices.
Thank you. One other thing is, or 2 things. One is, are you doing perfusion in these systems? And second question, how about the 3D environment when you make liver or kidney? Some of these things like gut is like a monolayer, but the liver and kidney, are they cells inside a gel type of environment or spheroid in a gel? Or can you explain a little bit more about the ECM cell things?
Yeah, definitely. I say the liver case, there are 4 different cells, right? So barrier, the bottoms, there are the endothelium cells, Cooper cells there, and the top is the stellate cell and hepatocyte cell and the ECMs. And there are, non-monolayers. They are 3D structures of models. So there is a very nicely signaling and not a monolayer based that. Okay. So TME is also important for the, TME is not only, one cell, but very different cells, right? So you can do some, different TME, and there are different cells, so you can check that efficacy by standard effect as well. Yeah, definitely not monolayers. It can be 3D structures.
Thank you so much. Well, that's all the questions I have. Thank you for your time, Professor.
Oh, great. Good questions. Yes.